Gene transfer to animal cells. We have discussed variety of techniques where exogenous DNA can be transferred into plant cells. And here we are going to discuss that how foreign DNA it can be transferred to animal cells. And first, briefly, we can focus on genetic modification of animals. Like the plant cells, animal cell culture technique has also been used to transform nuclear DNA, and it offers several advantages. Like in case of bacteria or other microbes like yeast or even higher plants, so sometimes problem is that they lack post-translational modification. That we have inserted our exogenous DNA, it has been expressed, and it has also resulted in the synthesis of a protein by the process of translation. But still, after expression, after protein synthesis, functional biological product it has not produced. All the product is there, but not functional. The reason is that most of the system they may lack post-translational modification, because post-translational modification they are very important for the biological activity. मिसाल के तौर पर बाज़ फंक्शनल ग्रुप्स एड्स होते हैं, मॉडिफाई होते हैं, रिमूव होते हैं, या प्रोटीन के अंदर बैंडिंग जो है वो इंट्रोड्यूस करवाई जाती है, जिसको हम प्रोटीन फोल्डिंग कहते हैं. तो ये एक सेपरेट टॉपिक है जो कि आप दूसरे सब्जेक्ट्स में भी इसको डिस्कस करें. बट हेयर वी कैन से दैट एनिमल सेल्स दे ऑफर one of the opportunity, and it is the post-translational modification of the expressed protein products. इसके अलावा on the commercial scale, animal cell lines that has been transformed, they can be used to produce some valuable end products. And here the production of antibodies is there, hormones, growth regulators, cytokines, vaccines has been produced by using animal cell lines that has been transformed with exogenous DNA. Now for its transformation, a variety of system has been used, and one of the system is viral mediated transformation. That foreign DNA it can be inserted as a whole genome in the phage particles, or even some genes can be replaced, and then it can be used to transform animal cell lines. But it is having one disadvantage, that is biosafety concern, that there are some phage particles they may be infectious and they can lead to the invasion of the host tissue and can cause infection. Because of this reason, some other techniques like particle bombardment, electroporation that we are going to discuss in next section has been used for animal cell culture transformation. So in first part, we will focus that somatic cell of animals they can be transformed. Somatic cells are those cells that are not going to transfer its genomic DNA into the progeny or the daughter cells, because they are more or less stable, and once they have been transformed with foreign gene, so they are not going to convert into transgenic lines. But transgenic lines can be established if we are going to target some specific cells of animals that are pluripotent or totipotent, that have the ability to transfer its genomic DNA into daughter or progeny cells. So here gametes, eggs, and even embryonic stem cells can be used for the transformation purpose, but this aspect we will discuss later on.